Welcome to Revolution Against Evolution. I'm your host, Doug Sharp, and I have with me a guest today. His name is Derek Marshall. He's been on our show several times already, and he's an engineer. You're a mechanical engineer, is that what you are?
More electrical engineer.
Or electrical engineer.
Mechanical, electrical, but currently working as a controls engineer, which is electrical design and programming of automated machinery.
Well, that's good, because what we want to do tonight is to hypothesize what it might take for us to build a machine to produce, mass-produce proteins. And there are such machines out there. And there's a machine called an amino acid analyzer, which will actually take a protein and break it down and figure out its structure based upon how the machine works. But I remember when I was in college learning about what it would take to synthesize a protein. And I think that it'd probably be a good idea to explain what a protein is first, so that you understand what the issue is and the problem. And a protein is a long chain of components. They're called amino acids. And what they do in the body, in life, are several different important functions. One is that of an enzyme. Another one is a hormone. A hormone is a protein. And so an enzyme sort of catalyzes reactions. It makes them go faster. And it actually latches onto a piece of food particle or something and breaks it down into simpler components. And so that's one of the things that I had a little bit of experience with enzymes myself.
I worked at Michigan State in the Biomass Conversion Institute, and what we did was we took stuff like corn stover and switchgrass, and we would break it down and try to convert it into ethanol. One of the things that we would have to use would be enzymes in a process called enzymatic hydrolysis, which was, just as you're explaining, it takes these larger molecules and breaks them down into singular molecules that could be further processed.
And the amazing thing that I learned when I was going through biochemistry was how the life actually produces proteins, because it's a rather interesting process. It's actually starts out with the DNA molecule. The DNA molecule is the famous double helix molecule. It has the computer code for life in it. And then there are three different types of RNA that are produced from the DNA. One is it's called the messenger RNA, which has the computer code for the protein. The other one is the transfer RNA, which is it sets up amino acids to be added to a protein chain. And then there's ribosome RNA, which the ribosome is is a container that is quite complicated, and what it does is it is the manufacturing process for the protein. And so the ribosome goes along on the messenger RNA. It'll take the three codons, and that determines what amino acid it's going to get. And there's a transfer RNA that fits on that, and it then adds the amino acid to the protein chain. Now, I discovered that when in my chemistry experiments, I decided, well, I was going to try to create life in a test tube.
This is the one I wanted to hear about again.
And it was a fun process, because I was enamored by the Miller-Urey experiment, where they were creating proteins by zapping hydrogen, methane, and ammonia in a water vapor. And it would produce these amino acids. And so I said, well, obviously, if life came about in this manner, then the next step is that you would form the peptide bond, which would combine two amino acids together and then have them come together in a growing protein chain, because I knew that this was needed for life to occur. Well, it doesn't work that way. The problem is that the components that were used to make the amino acids will conflict with that reaction. In particular, water. Water is a enemy to that reaction because you have to remove a water molecule to produce the peptide bond.
Something else you've told us about is the use of a right hand. There's a molecule that can only accept right-hand molecules.
All amino acids come except for one, glycine. Glycine is symmetrical, but all the 20 amino acids, 19 amino acids, are asymmetrical, which means they come in two different forms if you make them in the laboratory, like in the experiments I talked to you about. So, but life only uses the left-handed kind, and so I often give a little illustration of the protein factory that we're going to invent here. Has to produce produce proteins that are like 400 amino acids long at a rate of 400 tons a second. And we're going to have this protein factory in a race with a snail, and the snail has got a job of taking the entire Earth over to the other side of the universe and back, one molecule at a time. And he only moves at a rate of 1 millimeter in a million years.
Yeah. Well, I tell you what, 400 tons per second is way beyond what I was able to come up with. We're talking about grams or milligrams per run. It is, this is like a millionth of a metric ton.
Right, and so in any event, by the time that the universe fills up with all these proteins that we're making in our protein factory, we won't have a viable protein built by the protein factory if all we use is the mixture of the two different forms. You have to purify left-handed amino acids in order to make them work in the reaction. So that's problem number one, but we're sort of setting that aside as something that has already been solved. The chemists.
Well, the idea, just a couple seconds here, is that if you just randomly went out and got these amino acids, you're going to get half left hand and half right hand.
Right.
So for any random process that you're just going to throw together and try to create life, you have to deal with this 50% of right-handed that you can't use.
Mm-hmm, right. And if you get one of them wrong, well, the protein doesn't work. It doesn't do its job. Now, what's interesting is that each amino acid has its own little personality, and that's based upon its side chain. Now, there's the amine and the acid group that is what forms the chain of amino acids in the protein. And there's a simple protein called insulin, which has 51 of them. And so I took a look at what it might take to make insulin in the protein factory, and that's sort of stretching the limit of the capabilities of this automated process, which I found online. But what happens is that each amino acid has this side chain, and it either likes water or doesn't like water. It forms an ionic bond or a sulfide bridge or a hydrogen bond. A hydrogen bond is something else that is sort of a weak bond, or it's just, there's a force called the van der Waals force, which they really don't know what it does, but they know it causes things to sort of stick together. And so it's these forces that cause the protein to fold up in a ball of sorts, and and each of these side molecules affect the way the protein works as a catalyst. And so, like, it'll glom onto a piece of food particle, break it in half, and then if you get enough of the food breakdown, it'll bind to the other side of it and it'll stop the process. So it's a very controlled regulatory process that happens when the proteins are actually being used in the body. So we have I looked up this process. It says, what is a peptide? A simple introduction of peptide synthesis. And it talked about the problem of the water molecule. And that's why they have to have it done in what's called a solid phase.
Now, this is a cell-free method.
Yeah.
What we've been talking about for the first beginning of the show here is something that occurs in a tiny little human cell, which is on the micrometer scale, which is very, very small.
Right.
So now we're talking about a process. The solid phase is a cell-free process, meaning it's
Right.
just a chemical process done in the lab.
Yeah,
by
we're going to
hand.
build a machine with some intelligence in it so that we can actually get this to come about. And so the thing that I see happening with it is that it starts out with you have to cleave the alpha amino protecting group. And so there's a group on it that they actually protect it from reacting in a way that would compromise what you actually want it to do. And because these side chains also are reactive. Actually, they're more reactive than the carboxyl group, which is what you're trying to get to join to the amine group.
Which
so
the
it's
carboxyl group is very reactive. It's carbons and oxygen
right.
group is very, very highly reactive, electronegative.
And so then after you get that stage done, you have to wash it to remove that cleavage reagent, and then you couple the protected amino acid. You join the two together. You have to make it so that the reaction is such that it doesn't react with the rest of it. It'll react with this one particular thing. And so it's a little bit higher energy than these other reactions. And so they finally get it to join, and then you have to wash it again to remove the excess byproducts. So you got lots of inefficiencies built into this process. And so you might start out, I don't know how much you have of purified amino acids you have to start out with, but the yield for this really isn't that high.
Right.
And so this is the question that I have for you is, in order to do this, if you were asked to build the software to control this, what would you say would be
when I was at the biomass conversion lab, we worked with a chemical R&D process workstation that was built by ChemSpeed, and they're a company in Switzerland. And we set this up to pre-treat biomass. Mm-hmm. So we're talking about something that has pressure vessels. You can vary temperature, you can vary pressure. You can put something in a vacuum environment or a high pressure environment under nitrogen. You have a robotic arm in there where you can do pipetting, which is necessary. You're going to have to be able to do washing steps. Mm-hmm. You're going to have to be able to do extraction steps. So you got to be able to pipette, which is to basically like a squirt gun, squirt very precisely amounts of material inside of a pressure vessel. And you'll be able to also, after the reaction's done, you'll be able to monitor the reaction using a computer. And you'll be able to stepwise host the reaction by operating the robot, controlling temperature, pressure. If there's stirring involved, you can stir. You can have trays off to the side where you can get a certain reagent that you need. The robotic arm will come over, it'll extract the reagent, come back over to your vessel, squirt it in there, whatever you need, and then when you are done, you can take that mixture after it's done, and you can transfer it into another reaction or to another vessel which will host the next reaction. So this machine gives you a way to write code to automate a chemical process. Now, one of the challenges with something like we're talking about is you need a very sterile environment, because not only are you trying to keep unwanted reactions from happening, but you also don't want microbes or other forms of unwanted bacteria to be part of the reaction too, which will follow your reaction. Now there's some, now this auto plant had a lot of your regular we call chemical processes or chemical unit processes, which is a term in chemical engineering. But we could also talk about some of if you were to get the equipment by itself, the things that you need to do what we're talking about. We're talking about a PCR machine, which is something that you can use. It's a chemical cycling or temperature cycling little device that you put your DNA in there, your RNA in there, and it cycles the temperature so you can multiply it. And you can, it multiplies greatly, like 100,000 or a million times what you started out with. So you can take a small code or a small piece of DNA and you can make it, you can replicate it. It self-replicates. It hosts a self-replication. Also, another form of protein expression is called hosted, which you can use a bacterium to actually host the reaction for you. You can use what's called plasmids to bring in your code that you want to replicate. In that case, you need what's called electroporator, which is it's an item that takes the bacteria and you use electric charge to expand the outer portion of the bacteria so you can get your plasmids in there. You need centrifuges, you need incubators, you need ultrasonic cell disintegrators so you can get at what you want to get at. It does exactly what it says. If you have cells that you need to get what's inside, the raw material inside, you need these ultrasonic disintegrators. You need what's called gel electrophoresis to separate your proteins. So you're going to come out, when you have a reaction, you have a whole bunch of proteins there. There's only one that you really want.
That's right.
So you have to put it in this Western blot, they call it, and you put an electrical potential on this device, and the proteins that you want will all kind of like, they're all in a race. And the one that you want will come out and be the leader of the race, and you'll be able to separate it because the protein's going to go towards the positive charge.
I've used that process before. Chromatography is another similar process.
Yes, and that's the final one is either an HPLC or an FPLC, where it's another form of trying to get, you have a solid phase, you have a transport phase, and you have your test, your whatever your item is that you're actually testing. And the idea is molecules of different sizes are going to transport through this solid phase via the liquid phase, all at different rates. So you're able to separate and you can see them all, and you can pick out the one that you want to analyze. So these are all items that...
With the column chromatography, you have a
yeah, that's
tube.
what it's called, the column.
And you pour it down through there, and then it comes out the other end.
Right.
Separated. It's a separation technique.
You can do chromatography with a napkin. If you have a napkin, some alcohol, you can take a common ink from a ballpoint pen.
Right.
And say blue ink may actually have 3 different colors of ink. And you can see those colors creep. Since the different colors, the different molecule, the different size, it will transport along that liquid mobile phase at a different rate. So this is the way that we would separate out after we've made this, done this cell-free protein synthesis. We get a bunch of products. We get the unwanted products. We've hopefully protected the product that we wanted, and we want to separate it, and that's where you would use your column and your HPLC.
Of course, we're cheating in this process because we're actually using amino acids that have been extracted from life already.
Yes.
And so we're not explaining the origin of life with this process because we've already solved the problem with the left-handed amino acids by using the left-handed amino acids that we get from life. Now, and so, like I said, this is a question that evolutionists have not answered, and so they said they sort of set aside the abiogenesis thing and say, oh, that's not evolution. What evolution is that this is random mutations causing natural selection, and so, I think it's a little bit disingenuous to sort of set aside a major question. If you don't have abiogenesis, you don't have life. You don't have life at the start. If you don't have life at the start for evolution, what does it matter? Little fossils of whales that you line up in a row down the line, you haven't got to explain from the start.
That's true. Thought that I had on the way over here was we take for granted all the scales of basically we live at the scale of like 1 meter, where we're each probably 1.5 meters to 2 meters tall. Everything's at our same scale that we can touch, handle, and deal with. Life, what we're talking about, exists at a very, very small scale. These compounds that we're talking about, these molecules are in the micrometer scale and even smaller than that. Now, so that's another scale that we don't really have we haven't had much access to until relatively recently. So my question is, or I guess my comment would be that that scale of life does not exist on an island by itself. That scale has to assume, or at least depend upon, all the other scales that exist in the world being there in place, like something on the planetary scale, or processes that happen on our human scale, and also processes that happen at the atomic scale below that of the molecule.
And even on a macro scale,
right,
you need impressive quantities of this to explain what life, how life came about. You can't say, well, it just happened someplace over here, and you had a few molecules come together over here, and then they just started multiplying. No. You have to say, well, you need at least 410 proteins, I guess there was a figure that was given, to produce at least the smallest cell possible. And then I remember reading something with James Tour. He's a famous chemist that talks about the origin of life. He says, if you don't have a cell membrane, how can you explain DNA? And if you don't have DNA, how do you explain the cell membrane?
Right.
And so this is the same chicken and egg problem that we have with DNA itself. How do you have DNA without DNA polymerase? DNA polymerase is an enzyme produced by the process that produces proteins. And so if you don't have either of those, how do you get it all started? So the logical thing to presume then is that there is a creator involved in this whole process. He is the one who put this all together. He's the one who built it and put it into place. And it's actually pretty much the same process throughout all of life. And for a good reason, if you have food, it needs to be the same kind of stuff that life is made of. And so there's quite a bit of different things. So do you think you could build one of these for me?
Well, what we've been talking about occurs in a tiny little cell, and I was talking about the scale of things.
Yeah.
Now, the human body, the average human body, contains, let's say, 30 trillion, 20 to 30 trillion cells. Okay, and we exist, like I said before, on the 1 meter to 2 meter scale. Okay, so we're still, anything that I would make to build what you're asking me to build is going to be maybe 10 square meters of equipment,
Okay.
okay, at the very least.
So, fill a small room, huh?
Well, no, a lab. Yeah, it would have to be in a lab. It would be something that would take up probably 2 of these little studio areas here of the various equipment. If you wanted to do it piecewise, like I said, all this list of equipment, or my auto plant. The auto plant took up probably about twice the length of this rug here for that whole process. And even then, it would take me probably, I don't know, approximately an hour, not sure how long some of these reactions are, to create maybe a gram of usable protein. Like you said, the yields are relatively low, the efficiencies are relatively low. It would take me a very long time to create a ton of protein. I mean, that would be like a life, something that, it can be, basically, proteins that are created for use in labs are bought on the gram scale anyway. Say if you want to get some beta-glucosidase or something to help you break down biomass, you're going to buy grams of it, and it's relatively expensive to get that. But tons and tons of proteins, that's a very, very tall order. It would take me a long time, and it would take me I have 30 trillion protein factories inside my body that take up the space of maybe 2 square meters, whereas I can only produce, with 10 square meters, I can only produce maybe a gram in a couple hours, if I know what I'm doing, which I don't.
There's something like trying to produce a protein such as insulin would take months and months of planning. By quite a number of different scientists collaborating together to find out what exactly the sequence you would need in order to make the peptide bonds without actually destroying the side chains themselves. Actually, you have to take the side chains off, I guess, and then put them back on after the reaction is done. And so there's those side reactions that go on as well. And then, like, if they make one mistake during the process and leave out a step, well then you get a protein that doesn't work.
Yeah, I know a quick and dirty way to do protein. You can take a hay. This has been done by farmers for years. Take hay or silage and just stick it under a big tarp and spray a bunch of ammonia over it and let it sit for a couple days. Or we used to do it in the lab under a lot of time and pressure and temperature. That increases the crude protein content up to 10%, 5 to 10%. It also breaks the food, the hay or the silage down more, so it's more digestible, but it also increases the nutritional value of it. So because the ammonia is NH3, it's part of the amino acid can be made or from ammonia.
We got all these mean old acids we got to deal with.
Yes, the mean old acids.
And we'll see you next time on Revolution Against Evolution.